It's for me a pleasure to introduce you today to Alfredo Miñano. Alfredo got a degree in biochemistry by the Universidad Autónoma de Barcelona in the year 2000. Then he did a PhD also at the Universidad Autónoma de Barcelona in 2006. And from 2007 onwards, he's been a postdoc working in the laboratory of José Rodríguez Álvarez at CIBERNED and the Institute for Neuroscience in the Universidad Autónoma de Barcelona. He started then to work in neurobiology, mostly on the effects of A-beta peptides and oligomers on AMPA-type glutamate receptors. In 2012, he did an stay at the Robert Malenka's lab at Stanford University, and his general interests are focused on two main areas. The first one is the mechanism of synaptic dysfunction in Alzheimer's disease, and the second one is the identification of new targets and biomarkers to improve both Alzheimer's diagnosis and future therapies. His talk today is titled AKAPs proteins as therapeutic targets for early Alzheimer's disease. So Alfredo, thank you very much for your willingness to share with us your work today, and you have the floor. So please go ahead and share the screen with us. Thank you.
Thank you very much, Miguel. So I'm going to share the presentation. Okay, and well, first of all, good afternoon, everyone, and I would like to thank the CIBERNED Webinar Series organizer for giving me the opportunity to fill you in one of the projects that we are developing in the lab about the rising role of AKAPs as a potential therapeutic target to early Alzheimer's disease. As you already probably know, Alzheimer's disease is the most important neurodegenerative disorder with a long process of brain degeneration, represented here in these different stages. The pathology faces several problems, but according to this image, one of them is the lack of good experimental approaches to give effective treatments because sometimes it's too late. We put our eyes on the effect of soluble forms of amyloid beta on synaptic function, the very beginning of the pathology when synapses collapse. If we increase our efforts in deciphering what is happening here, surely we would increase the number of Alzheimer's drugs in development related to synaptic targets compared with the current pipeline. Thus, a real need exists to progress toward new drug development, and we believe the synapse should gain in value. Effective treatments for AD needs to target its preclinical stage known as cellular phase. In early phases of AD, learning and memory processes are mainly altered, involving many characters. LTP is impaired, LTD is potentiated, where AMPA receptor trafficking deregulation plays a pivotal role. On this deregulation, we put the focus on AKAP 150, a key signal scaffold protein. In parallel, we know that synaptic function is a high dynamic and bioenergetic demand process, where mitochondrial network is affected in AD. Here we put the focus on the other mitochondrial membrane AKAP that coordinate fusion and fission events altered in AD, leading to the bioenergetic failure. Synaptic AKAP 79 in human and 150 in rodent is a postsynaptic scaffold protein that anchors PKA, PKC, and calcineurin to the PSD, regulating AMPA receptor phosphorylation, trafficking, and activity associated with LTP and LTD processes. Moreover, AKAP has emerged as a key coordinator of L-type calcium channel signaling in the nervous system, an important function when considering the roles that this channel play in controlling neuronal plasticity, development, and gene expression, in addition to the well-established role of calcium dehomeostasis in neurological disorders like AD. However, it's still unknown whether degradation of AKAP is triggered by oligomers and whether it could be associated to AMPA receptors endocytosis. On the other hand, the AKAP1 or AKAP 121 in mouse or 149 in human primarily localized into the outer mitochondrial membrane, where it scaffolds kinases, phosphatases, and other signaling molecules that regulate mitochondrial physiology and health. Anchoring pKa coordinates mitochondrial fission and fusion events, essential for regulation of mitochondrial function. And furthermore, it's still unknown whether the degradation of this AKAP is also triggered by oligomers and whether it could be associated to the deregulation of the fusion events that occurs in AD. So synaptic dysfunction and mitochondrial breakdown are then two of the main contributors to neuronal death in AD, and it is vital to know whether AKAPs are involved in the molecular causes that trigger the disease at synaptic level, early stages of AD, to find a better and earlier therapeutic approach. Before launching our hypothesis, we check the levels of both AKAPs on AD brain human samples, and we observe a clear reduction in their levels and direct correlation between changes in synaptic AKAP and changes in synaptic proteins, indicating that the protein is affected in the pathology, and as a consequence, their signalosomes are related with functions shown in the previous slides. So our goal in the lab is to demonstrate the pivotal role of AKAP degradation in AD, induced synaptic and metabolic dysfunction, and exploiting them as platforms for precision pharmacology. Today, I will share with you briefly some results regarding the mechanisms linking synaptic AKAP degradation and synaptic dysfunction affecting AMP receptor trafficking and expression that may occur at early stages of Alzheimer's disease. So it is well established that LTD and beta oligomers share mechanisms of action on synapses, as you can see here in this image, and we report several years ago that oligomers were able to induce a calcium influx through ionotropic glutamate receptors, triggering GLUE1 dephosphorylation in the serine 845, and AMPA receptor. This is a serine that is related with the pKa and calcineurin function in the regulation of these AMPA receptors, and AMPA receptor endocytosis in a calcineurin-dependent manner. So recently, studying the same mechanisms, but related to NMDA-dependent chemical LTD, the reduction in the phosphorylation, in the same phosphorylation, and GLUE1 levels, together with less surface expression of these receptors, the membrane, we observed that the key scaffold protein that regulates pKa and calcineurin function, the AKAP 150, become degraded around the time. So we decided to study whether the oligomers were able to induce AKAP degradation by the same mechanisms. And effectively, we observed that oligomers triggered AKAP degradation through ionotropic glutamate receptors and involving the ubiquitin proteasome system. So we are working on restoring the AKAP levels in presence of oligomers and see what happened to AMPA receptor function, as we previously reported a direct relationship between the presence and the absence of AKAP and the expression of GLUE1. Then we will decipher the importance of AKAP on the short-term effects on AMPA receptor trafficking and synaptic function in early Alzheimer's disease. Once we observe this relationship between AKAP and GLUE1 by oligomers, we wonder whether AKAP was also affected by oligomers, but not only at short exposure, but also chronically, becoming a key protein in early Alzheimer's disease. We observe in this chronic or this stimuli during the long period of time a slight toxicity of oligomers in culture, and that prolonged absence of AKAP affect GLUE1 at long term at the synapse, the presence of the synapse, by affecting the protein. See with this here in this image, we can see that the levels of AKAP remains less or diminished during a period of time, a long period of time, and then we observe a decrease in the expression of GLUE1 in the amount of the protein of the GLUE1 affecting the protein, and also we observe, checking the expression of the messenger, a decrease in the gene expression. So then we decided to explore AKAPs silencing as a model of chronic absence of AKAP to see what happened. Using a shRNA-mediated acute knockdown of endogenous AKAP, we were able to effectively reduce this AKAP expression in cultured neurons. This knockdown of endogenous AKAP did not induce significant changes in other scaffold proteins like PSD95 or SAP97. However, this AKAP silencing decreased GLUI1 phosphorylation and total levels of GLUI1. Consistent with the results, AKAP knockdown reduced both synaptic and dendritic GLUI1 levels, followed by a decrease in the surface GLUI1 expression. So considering that AKAP is able to modulate gene expression by regulating NFAT function, we check whether the GLUI1 promoter had consensus recognition sequences for NFAT family members. We found several sites, and we cloned a fragment of the promoter with these sites on a luciferase reporter. The analysis of the GLUI1 promoter activity showed us that both the presence of oligomers in a dose and time-dependent manner and knocking down AKAP affects GLUI1 gene expression. And moreover, we start with a preliminary analysis of NFAT4 location and NFAT transcription factor activity dependent of the luciferase assay. And those preliminary results show that AKAP is necessary for NFAT translocation to nucleus, for example, under bicuculin treatment or with depolarizing stimuli with KCL, as it has been previously reported. When AKAP is missing, there is no increase in the translocation to the nucleus after the treatment. So you see here, for example, most clear effect of the depolarization with KCL, translocation of NFAT to the nucleus is really important. There's a high increase in this translocation. But when AKAP is missing, the amount of NFAT that translocates to nucleus remains more or less constant. There is no increase, or the NFAT4 is not able to translocate to nucleus. And also, what we observed was that it seems that it is NFAT specific because when we silence AKAP, we observe a decrease in the activity of the promoter related to the NFAT transcription factor. So recently, there is a growing evidence showing the important roles of several transcription factors in the communication between synapse to nucleus. So despite this recent paper that described a possible mechanism about how the activation of the synapse trigger NFAT transcription factor function, mainly through L-type calcium channel and calcium spike propagation to the soma to activate the calcineurin in the soma and then the phosphorylate and translocate to the nucleus, we decided to explore whether NFAT4 could be located at dendrites, considering the location of AKAP at spines and its important function regulating pKa and calcineurin NFAT pathway. And our preliminary data of NFAT dendritic location and potential mobilization away revealed that bicuculin treatment was able to reduce the dendritic NFAT4. Here you can see here that the bicuculin treatment reduced the dendritic NFAT in the dendrites, moving, or we suppose, to mobilization from the dendrites to the soma. Moreover, the lack of AKAP showed reduced levels of dendritic NFAT4 just in basal conditions, indicating how important could be the presence of AKAP in the spines for the correct communication between the synapse and the nucleus. So further experiments that we are performing are needed to confirm this possibility. Despite that, as I mentioned before, several reports show that mainly effect of NFAT is on the soma or through the activation of calcineurin NFAT pathway on the soma, and not communication between the spine or each individual spine to the nucleus directly. So finally, we decide to explore the mechanism that regulates NFAT translocation from cytosol to nucleus. So this translocation depends on its phosphorylation levels. When MFAT is phosphorylated, it is retained on the cytosol, but when MFAT is dephosphorylated, mainly by calcineurin action, then it translocates to the nucleus. So we see here clearly the mechanism of the regulation of these transcription factors. So we explore the phosphorylation levels of MFAT4 and its nuclear translocation and their chemical LTD condition or in presence of beta oligomers. And we effectively observe an increase in the phosphorylation levels after the treatments that correlates with a reduction in the MFAT4 nuclear levels. So here, this increase in this phosphorylation that depends on the calcineurin and pKa regulation of this transcription factor, and the increase in the phosphorylation after the chemical LTD that it correlates with a decrease or a reduction in the translocation of this transcription factor to the nucleus, here in the LTD and also in the presence of oligomers of amyloid beta. So summarizing this part of the work, we are trying to decipher the mechanisms by which the degradation of AKAP induced by oligomers could be disrupting synaptic function. So the short terms that we have seen related with the AKAP degradation via a proteasome system, the regulating AMPA receptor trafficking affecting synaptic communication, this should be these short-term effects in the local trafficking of the AMPA receptors at the synapses. And on the other hand, we have seen how chronic exposition to oligomers prolong the absence of AKAP, affecting the expression of new GLUI1 subunit of AMPA receptors that are necessary for the correct synaptic function. We have observed that the absence of AKAP deregulates the pKa calcineurin MFAT pathway, so the MFAT4 remains phosphorylated and is not able to translocate to the nucleus and then activate the expression of the GLUI1 subunit, the gene. So in this context, we saw a reduced expression of GLUI1 subunit having the consequences in the excitatory neurotransmitters. So together, participating in the synaptic dysfunction that appears at early stages of Alzheimer's disease. So our future goal in the lab now is to decipher in deep the pivotal role of AKAP degradation in AD-induced synaptic and metabolic dysfunction. So in this way, we are starting to work in this other AKAP, this AKAP that is this mitochondrial AKAP that share the same actors that we saw previously in the synaptic AKAP, the pKa and calcineurin, and how the degradation of this protein could be affecting the function of the mitochondria, the fission or the regulation of the fusion and the fission processes that we know that happens in AD, with the idea of that this AKAP, the synaptic AKAP and the mitochondrial AKAP share regions or homology regions that are susceptible to be ubiquitinated and degraded, with the idea of trying to decipher if these oligomers are able to, in the same way that happens in the synaptic AKAP, degrade this protein with the same mechanism, with the idea of target to open this computational framework in which by bioinformatics we can play with these possible molecules that could, in this case, for example, this is a process in which we can induce the ubiquitination of proteins, adding tags of proteins or to specific targets. But in the same way, we will be able, by pharmacology, try to avoid the degradation of these proteins in the context of the pathology, trying to stabilize with these studies of the pharmacology, stabilize the AKAP signalosome, restoring the crucial function for neurons at early stages of Alzheimer's disease. So the idea was would be using AD patient somatic cells and then transforming that in iPS cells, and then using these relevant cells derived from a disease as a model for this screening of drugs according to the data of the bioinformatic information. So we would do this screening with these drugs, and then we'll try to decipher if AKAP could be this new potential therapeutic target to Alzheimer's disease. So our take-home message today in this, I guess this short talk, but I think that is important to open the time for the discussion of how these AKAPs are emerging as a key proteins in early synapse dysfunction, and they are a promising potential therapeutic target in Alzheimer's disease. And finally, we would like to acknowledge the past and present lab members, our collaborators, and the institution for funding. And thank you very much for your attention.
All right, thank you very much, Alfredo, for a nice talk. So now the paper is open for discussion. As I said earlier, if anyone is interested in making a question to Alfredo, please do so by raising your hand using the application option, or by sending me a message through the chat option, indicating that you're interested in making a question, and just stating your name. So I will give you the floor for make the question yourself. So while we wait for the first question, let me remind everyone the CIBERNED Webinar Series is held every other week on Thursdays at 3 p.m. And if you're interested either in participating or looking at the future programs, do so by checking the CIBERNED website for the program, and also to get in touch with Teresa Iglesias, with the coordinator of training in CIBERNED, and in charge of arranging the future webinars for the next few months. Okay, so I don't see any raised hands or any message through the chat coming in for questions. So let me remind also everyone that the seminar is being recorded, and the recording will be available in CIBERNED website in the next few days. So we have a first question by Carlos Saura. So Carlos, please go ahead.
Yes, thank you, Alfredo, for this nice talk. It's very interesting that you find these changes in AKAP in eddy brain and in your culture conditions and treatment with beta amyloid oligomers. I want to know whether you have checked also whether other AKAP proteins are affected in the brain or in these cell culture conditions, and also you have really tested the effect of beta oligomers on AKAP levels and GLUR1, but what is if any, the role of tau pathology in the regulation of AKAPs?
So we check the levels of the mitochondrial AKAP on 80 brains, human samples. That is what I show you in the presentation. and it correlates perfectly with the decrease in the synaptic AKAP and the decrease in the mitochondrial AKAP in these 80 human brain samples. I didn't check any other kind of AKAPs that we can find abroad, but well, I consider so, and this is the reason by which I focus on these two main AKAPs or members of the family, because the importance of the two functions in the synaptic function, so the synaptic communication and the bioenergetics of the high demand of energy that the neurons have. So, and then considering that these AKAPs share domains that are conserved, and both proteins seems to be regulated by mechanism of ubiquitination. So just this is the other part that we are starting now. So trying to check whether these oligomers are degrading the mitochondrial AKAP as it happens in the synaptic AKAP, and see if the mechanism by which these oligomers share these effects on both, with the idea to check whether these AKAPs could be this therapeutic approach. So what happened with the tau pathology, at least we have not we did not do any we didn't do any kind of experiment to check whether tau or the part of the tau pathology could be related with the AKAPs. I don't know in how which is the relationship that you can find between this tau pathology and AKAP.
Basically, because tau is deregulated and also accumulate at dendrite or synaptic regions during pathology. So maybe that way pathological tau is somehow disrupting the complex of AKAP with other partners there in the dendritic spines, for instance.
Yeah, maybe we can try to check. So the idea is so knowing which is the molecular mechanisms that affect or disrupt this pathway of this complex signalosome between PKA, calcineurin, and AKAP in regulating trafficking of the receptors, but also what is happening with AKAP regulating these cell-type calcium channels and the activity. So we have different kind of ways to explore the complex. Also because considering that we don't know if this regulation of MFAT is produced in the soma, only in the soma, as we have seen in the paper that talk about the communication between the spines and the nucleus, or maybe these signalosomes are important in the dendrites, because if AKAP is important regulating the trafficking of the receptors, and it could be important for the message that is happening there to go to the nucleus, we have to explore all the possibilities. And why not this the accumulation of proteins on the regulation of all the processes that happen related with tau maybe could be related with what is happening there in all of the actors that are in the synapse. So we will explore if something is happening related with tau.
Maybe it's just to follow your answer just very briefly. You know that there are several factors that at least in our lab we have seen that synaptonuclear factors that are affected during the pathology. So have you tested whether AKAP or the regulation of AKAP can also affect these other synaptonuclear factors and the nuclear translocation of these factors? Or?
Not yet, because for us it was a surprise that to find a relationship between the lack of AKAP and changes in the expression of an AMPA receptor subunit like GLUE1. So it's like when AKAP is missing, something is happening in this short term related with the trafficking of the protein, but something else happens in a long time related with the expression of this protein. So I don't know if the lack of this AKAP could be affecting other transcription factor function, for example, CSTC1, because at the end, the regulators of these transcription factors are common, that is the calcineurin phosphatase. So maybe and this is one of the important, I guess, for us, one of the important point of AKAP as a therapeutic target is that AKAP is very upstream of what is happening in neurons. So it seems that it's a key scaffold regulating protein that could be acting through this AKAP at the very beginning of the activation of the protein, or what is happening after the receptors. What is happening there downstream open a lot of possibilities that this upstream protein could be interesting to target it pharmacologically.
Okay, thank you. Yeah, will be interesting to see if you can target the AKAP finally as a therapeutic approach.
Maybe in future.
Thanks.
We hope so.
All right, thank you, Carlos and Alfredo. Any other questions? No, I don't see any raised hands or messages coming through the chat. Okay, so I have a very short question, which is out of curiosity, Alfredo. Are AKAPs detectable in CSF? Thinking in terms of biomarkers.
I don't know. I guess this is a very big protein, but well, sometimes this is not a problem to find the protein at CSF. But I have not checked it if it's detected or not in CSF. So it could be interesting to check whether this protein could be
Yeah, because you mentioned a couple of times during your talk on the potential use of biomarkers of this pathway. So are you thinking on the AKAP itself, or more downstream elements that could be affected by AKAPs?
So like biomarkers. So when we thought about AKAPs as a therapeutic target, is just thinking in how we can modulate the stabilization of the signalosomes, thinking in the function of the neurons. So when we refer to biomarkers, is because in other project that we are developing in the lab was more related with microRNAs as modulators or regulators of the expression of proteins that could be interesting in the context of the pathology. And of course, microRNAs are found in the CSF and also in plasma, and they are very conserved, and they are a really good possibility to use them as a biomarkers for the early diagnosis of Alzheimer's disease. And of course, we know that there are some microRNAs that target AKAP that could be, why not, releases to the plasma because of the regulation of. So we know that A beta is degrading the protein, but we don't know if there are other kind of changes in the expression of microRNAs that regulate the expression of that protein that could be also adding more importance to this protein and the possible pathway related with this. So maybe there are some microRNAs that attach or that regulate the expression of the AKAP that could be deregulated, and we can find it or them in the plasma or in the CSF. So these are these two different projects or that we are developing in the lab. So find therapeutic targets by itself acting directly on the protein, on stabilizing the protein, and the other is molecules that could be regulating the protein that could could be detected like a biomarker.
Okay. All right, thank you. So again, so if are there any questions left? Again, I don't see anything coming out of the chat or any raised hand. Okay. So, okay, so if there are no more questions, I think it's time to close the session. But not before thanking again Alfredo for a nice talk and for his willingness to share with us his work. And thank you all of you for attending. And I wish you all of you a nice rest of the day. I'll see you in the next webinar. Bye-bye.
Bye-bye. Thank you very much.
Thank you.